Welcome back friends, welcome to another lecture from Just Biology. In this lecture we're going to talk about a molecular biology. When we talk about molecular biology, we have to know a certain process. Apart from the metabolism inside the cell, we can understand or will come to know there are some process takes place inside the cell which is important for the information so that can be transferred to the next generation. Those processes can be referred as a fundamentals of molecular biology. So better to say they are the central dogma of the molecular biology. So what do you mean by central dogma? So that we'll discuss first, then we'll move on to the each and every steps inside the central dogma. So let's begin with DNA. So we know that the DNA will undergo a certain process. That process will give you the RNA molecule. So that process will give you the RNA molecule, and obviously this RNA will be converted or they are transformed into the certain things or polypeptide. We can call it as protein. We can call it as protein. So this can be referred as the flow of the genetic information which is transferred to the next generation. So this can be referred as a fundamentals of molecular biology or central dogma of molecular biology. So this is what we are going to discuss now. Let's begin with the conversion of DNA to RNA. A DNA which is converted, or they are actually the one of their stand will be acts as a template for the synthesis of RNA molecule, and this process also specific, and so that happens only when need the information whenever they need to multiply. So actually this process will be occurs during meiosis stage only, because where only the information will transfer until or unless it remain as such. So that is the idea. Now this DNA will be converted into the RNA, as I said before. So what we can call it as this process? No, everyone will know the process. Let's say, so this is called a transcription. Transcription. Further, this RNA will be converted into the protein by the process. We can call it as translation. The process will be referred as translation. So there are many differences between transcription as well as the translation that we'll also discuss in future lecture. And now just remember one thing, the DNA which is transcribed into the RNA. Now, so my question is, if you take prokaryotes and if you take eukaryotes, whether they possess same? The answer is no, because the prokaryotes don't have any unwanted sequence or the non-coding sequence that referred as introns. They lack introns. But in case of eukaryotes, you can see a many numbers of repeated sequence that is referred as an intron. We have the intron, but in case of prokaryotes, they have no introns. So their gene is not interrupted, or their coding gene is not interrupted at all. Now, the transcription of these prokaryotes will be different from the eukaryotes. So that we'll discuss in a future lecture, or upcoming lecture we'll discuss each and every steps in detail. Now let's look at the DNA. The DNA is double stranded molecule which packed inside the nucleus. Remember, this DNA molecule should multiply, or we need a copy. We need many copies of DNA molecule. Once they undergone translation or transcription, whatever process they are going to start with, we need many copies of DNA. Now my question is, how do we get the many copies of DNA? there is a process which is referred as replication. The replication also be a part of a central dogma of molecular biology. Let's write it here, central dogma. So we can abbreviate it as CD, central dogma of molecular biology. Now, they also have a specific function, so that is called replication. Actually, the replication is a process, the DNA which can be duplicated. The replication, which is a process by which the DNA will multiply themselves with the help of many enzymes or the factors. Now, the technology has grown enough. Now, the scientists were discovered a certain technique which we can make DNA from the mRNA molecule. So that is just reverse of transcription process, just reverse transcription, the reverse process of the transcription. So we can refer it as a reverse transcription here, reverse transcription, and in which we can synthesize a full-fledged DNA of a particular RNA. From the use of the RNA, we can make a DNA molecule, so which is coded for. And this process is referred as a reverse transcription, and the enzyme which involves inside the reverse transcription is reverse transcriptase, and this process takes place only inside the viral particle. So in between DNA and the RNA synthesis, there are some process unlike prokaryotes. If you take eukaryotes, as I said before, they have non-coding sequences. So we don't need any kind of those introns, then we need to remove. And also we need to make some kind of modification to activate the RNA molecule. And those processes will be referred as post-transcriptional modification. Post-transcriptional modification, we can call it as post-transcriptional modification. Why? Because this process takes place once the transcription is getting over. So that's why we can call it as post-transcriptional modification. On the other hand, the same way you can see after the translation process, we can also see a same kind of process after the translation. That process we can call it as post-translational modification. Post-translational modification. So simple. The idea is simple. Because RNA which is synthesized after the transcription is in inactive form, we need to tap it because we need to activate them. For that we use post-transcriptional modification. In post-transcriptional modification, we have three different processes that processes are very specific to at each and every organism. The first process, you can see this post-transcriptional modification is splicing. And the second process is capping. The third process is adenylation. So this is the three post-transcriptional modification process. Let's begin with the splicing. The splicing means the process which remove or which the RNA which completely refrained from the non-coding sequence, or that is called introns. What are all the things we need, we'll take those information alone and the rest of the thing will be removed. So that's why the RNA will undergo splicing. Now let's come to the capping. What is capping actually? Just cap. For example, this is mRNA. We can make a cap over here. Why? Because we need to protect the end of the mRNA. Why? Because the 5' phosphor, actually before discussing capping, so we should know the structure of mRNA. So I'll draw here. So this is a rough drawing. So let's imagine this is mRNA, and this dot, this dark spot will be cap, and this is actually this is spliced one. And the third process I said adenylation. So I want to discuss these three topics in a single picture. Let's say this is pollination, this is mRNA. Now if you see the cap of the mRNA, now this is a 5' end of mRNA, and this is probably 3' end. If you see the 5' end of the mRNA, you could see a lots of guanine residue over there. That is important here. Why? Because this guanine residue is more stable than any other nitrogenous bases. Then, so rather than actually the addition of this guanine base is important here. And if you add some sort of let's say some hydrocarbon group over the guanine molecule, so that becomes inert. So that become inert. They cannot react with unwanted chemicals or unwanted group of things or unwanted group of protein, whatever else. The cap will be bound with a molecule of CH3. So that is methyl. So this cap will be referred as a methylated guanosine cap. Why methylated? Because this methyl group will be present over the guanosine. So this is probably a guanine molecule, and the guanine molecule will be bound with CH3. So that is methane. So methylated guanosine cap. If this capping process will be over, so this DNA or this is RNA will become inert, or they cannot react no longer. So that's why this capping process is important here. Let's look at the third process, that is adenylation. The adenylation, this is not an adenylation, this is polyadenylation. Poly, because you can see a number of structure adenine group or adenine molecule or adenine base you can see at the 3' end of the mRNA. And what is the function of this adenine? Actually, this adenine has a different dimension. While we are talking like purification process or some kind of identification process, we can use this adenine residue for identification for that purpose. Apart from those things, this mRNA should have a certain lifespan, and they should have some, they have some sort of span. Within that time, the mRNA will synthesize a sufficient amount of protein which we required, and the mRNA will be added with, let's say, 20 to 30 adenine residue. Let's imagine. So this has a particular lifespan, and the mRNA will have 50 to 100. They have comparatively a more lifespan than 20 to 30. So that's why these adenine residue, which represent the lifespan or the duration of the mRNA, or they are capable of synthesizing the protein. So that is the important process here, because most important. Why? Because that information will be reduced, or the whatever things we needed, that will only taken by the mRNA. That mRNA will converted into the protein, and the protein will be secreted. Once they secreted the protein should undergo the same process like post-translational modification. Why? Because they will have a certain modification. They need some modification for activation of protein. Let's say, so this is, we can refer as a polypeptide. So let's erase here. So let's imagine this is a polypeptide. Now, so once the translation is getting over, the protein which is translated from this RNA is inactive form. Why? Because they are immature, or they don't know what they need to do, or what it does actually. For that, we have a certain modification. We have many things actually. The protein that includes hormone, enzymes, some sort of factors, or a transcription factor, or a regulatory protein, and so on. Many proteins we have, and this protein doesn't know themselves which which kind of protein they are. For that, they need some indication, or they need some tag. That tag will be added, or the naming given after the only after post translation modification. Whether we can differentiate hormone or enzymes or whatever it may be, only the post translation modification only we can identify. That's why these two processes will be important. Now, so as I said, they are immature. How the protein will become mature? The same way, the unwanted portion of the RNA, or this mRNA will also spliced. The same way, the unwanted peptide or the unwanted amino acid should also spliced in case of protein. Then the signal peptide, in case of hormone, it will be attached with, or it will be bind with a certain peptide is called a signal peptide. So what is the function of actually the signal peptide? The signal peptide will receive the signal from the site of protein requirement. So that requirement will be assigned for the signal peptide. The signal peptide will go and try. This signal peptide will bring this protein to the site of work, and the work will be done by the protein, and the signal peptide will be cleaved off. So this is the basic idea of the central dogma of molecular biology. Now, in upcoming lecture, we'll be discuss each and every process in detail. I hope you like the video, and if you like the video, please hit the like button, and don't forget to subscribe my channel and click the bell icon for latest updates. Keep watching guys, and stay tuned.